Welcome everyone to the next iteration of the IITE seminar series. Today it is Jonas Wickman who is going to be talking to us. He's a postdoc at Michigan State University, and he's going to be talking about novel methods for modeling eco-evolutionary dynamics, and with a special attention to modeling the evolution of the trait variants of those species. So without further ado, Jonas, the floor is yours.
Okay, thank you very much. So this is work I've been doing with my postdoc advisor, Chris Klausmeier, and Thomas Coffel, who also until recently was a postdoc of Chris's, but who is now an assistant professor at University of Lyon. So the last decade and change, there's been this sort of explosion of literature on intraspecific trait variation, especially in the empirical literature. And there have sort of been, broadly speaking, two sort of large furrows that have been sort of plowed scientifically there. And one of them is just how is trait variation apportioned among organisms? So is it intraspecific, where, for instance, we have like a short blue tree and a tall blue tree, but also a short orange tree and a tall orange tree? Or is trait variation more interspecific, where we have short blue trees and tall orange trees? And if there is intraspecific variation, how is that sort of apportioned in the landscape? Is there local within patch variation, where the blue trees are both tall and short in all patches? Or is there primarily between patch variations where the blue trees are short in patch 1 and tall in patch 2? And the second big question that is being asked is, what is the effect of ITV on higher order things like, does it help or hinder species coexistence, and what does it do to ecosystem functioning? But I think to a certain extent, like a more practical question for these researchers underlie all of this, and that is like, how necessary is it to include ITV to understand your study system or to answer scientific questions about your study system? And my training and expertise is in eco-evolutionary dynamics. And when I see a distribution of heritable traits that potentially affect fitness, what I think is like, oh, there's going to be some eco-evolutionary dynamics. So if we sort of consider some habitat or some region, then we're going to have evolution through natural selection and mutation acting, and that's going to change the traits over time. And when the traits change, it's going to change the ecological dynamics in the system, mediated, for instance, by consumption or competition. And when the ecological dynamics change, it changes the environment experienced by each individual. So the number of resources or amount of resources changes, the other individuals you interact with change. And that sort of, once again, feeds back into the evolution through changing the selective environment. And on top of all of this, there might sort of be unused trait space in some sense. So new species might come in and invade the system and add themselves to the community, or there might be evolutionary branching, where one species splits into two over time due to selective pressures. And sort of this feedback loop goes around and around, but in the very long run, we can expect to reach some kind of attractor in the simplest case, and some kind of equilibrium where we have no changes in traits, no changes in densities, and no changes in the environment, and where the community is closed to further innovation, and consequently also no more evolutionary branching. And because these these communities serve as important attractors for the systems, they've been given the name evolutionarily stable communities, and sort of studying their properties is important as they serve as sort of the long-term end goal of these dynamics. So then the question is, in these stable communities, how will trait variation be distributed? So we're going to sort of look at two broad ecological questions here. And the first one is, what happens to trait distributions when selection goes from strongly stabilizing to weakly stabilizing or disruptive? And then similarly, but on a regional scale, is what happens to trait distributions when spatial conditions become more heterogeneous and different from point to point. So to sort of illustrate the first question is a little bit more in detail, is that sort of when we have strong stabilizing selection, we can imagine that we would get an outcome close to this. So we have some trait X, and then we have a distribution within a species of a trait density distribution. And if the selection is very strongly stabilizing around an optimal trait 0, then we would expect a very narrow trait distribution like this. But under different conditions where selection is not strongly stabilizing, we could imagine that we either get something like this, like one very wide trait distribution, Or we could imagine that we can cram a bunch of species into the same environment, but where each species has a very narrow trait distribution. Or, of course, some kind of combination of these two outcomes, which is probably the most realistic. And in space, sort of similar things play out, especially with regards to local adaptation. We can look at a toy example where we have some density distribution over a spatial coordinate now, a spatial coordinate Z, and where the sort of this cartoon model has some local growth and random dispersal. So we have some density profile over space where the spatial coordinate could be like the depth of a water column or how far up a mountain you are, say. And if you don't have any intraspecific variation and all individuals are identical, then you will get something like this because all the individuals are identical. There's also no variation in space. But if you do have some variation in space, then this has sort of the consequence due to the dispersal that for any focal point, you will sort of have guys coming in from up the mountain and guys coming in from down the mountain. And if their traits are different, you will sort of mix these different traits. And the consequence is that you will have intraspecific variations. There will be some variation around the mean at each point. So this local adaptation begets intraspecific variation. And then similar to the non-spatial system, we can imagine that we could potentially have a set of different outcomes. So we could either have this sort of outcome depicted in gray, where we have few species with a lot of local adaptation. We could have a lot of species depicted here as dashed lines with basically no local adaptation or intraspecific variation. Or we could have some intermediate outcome as depicted here in blue and orange. So, sort of what theory do we have to try to tackle these questions? Well, I would say we have sort of two broad streaks of theory. And one is what I'm calling distribution and moment-based methods. And these come in a lot of different sort of flares. You've got quantitative genetics. There's a sort of community ecology framework. There's something called trait diffusion approaches and something called oligomorphic dynamics. All of these are sort of closely related formalisms, and they're all based around tracking these distributions or often tracking these sort of mean traits X bar and trait variances V for each species you're interested in. So on the other hand, you have adaptive dynamics. And in adaptive dynamics, you have no intraspecific variation whatsoever. But what you get in return is that you have sort of a very mature theory for assembling a stable community by evolutionary branching and using invasion analysis. And you can really sort of tell at the end mathematically when you're done assembling a community. So these two frameworks have complementary strengths and weaknesses. In one, over here, you can include intraspecific trait variation, but you don't really have the tools to perform this kind of community assembly. And in the other, you do have these tools for community assembly, but by construction, you have no intraspecific variations. So you can't really address these questions I proposed. So in this talk, I'm going to introduce a framework we've come up with for trying to combine these two facets of eco-evolutionary theory. And we're going to study question one, what happens as selection becomes less stabilizing in a non-structured model. And we're going to use a simple Lotka-Volterra competition model to sort of try to get at this question a little bit. And then for question two, we're going to extend this Lotka-Volterra competition model to a two-patch model to see what happens when conditions become more heterogeneous. All right. So the basic object of study we start with is this trait density distribution. So we have some trait, and then we have a trait density distribution that describes how many individuals there are for each trait. And then we can formulate something we're calling trait space equations that tell us how this trait density distribution will change over time. And basically, these equations have three ingredients. They have a birth rate B, a mortality rate M, and both of these rates can depend both on the focal trait of the individual X, but it could also potentially depend on the entire trait density distribution in the community. And the final ingredient is mutation. So we're convolving this birth rate here with a normal mutation kernel. And what this means is that while we do assume clonal dynamics, we assume that you don't give birth to perfect clones of yourself, and instead each parents give birth to sort of a normal distribution of offspring with some mutation variance, capital M. And then, similar to most other frameworks, we're not really reinventing the wheel here. We're going to assume that we can decompose this trait density distribution into a number of pseudo-species, perhaps one for every peak here. And then we're going to approximate each of these by a normal distribution. So we're going to, for each species, we're going to keep track of U, the total density, the area under the curve for each of these. We're going to keep track of the mean trait, X bar, and we're going to keep track of the trait variance, V. And because we're assuming normal distributions, these three numbers are enough to completely characterize each species. And then V tilde is just going to be the sum of all of these approximate trait density distributions. And before we can formulate the equations for these three moments, a little bit of notation. So we're going to write B hat and M hat for the population level rates. And that basically means that we're integrating, for say, the birth rate over an entire normal distribution. So for example, if you have an exponential function, as we do here in green, B equals e to the alpha x, then for each of these species, we sort of have to integrate this rate against this normal distribution, and you'll end up with a population level rate that depends both on the mean X bar and on the trait variance of the population V. And now we can sort of formulate equations for how these three moments, the total density, mean trait, and variance, change over time. And these are very similar if you ever to other frameworks, if you've ever worked with any of them. So the density for each species is simply going to be the per capita population level birth minus mortality times the total density. The mean trait is going to change according to a sort of a Landé or canonical equation type equation, where you have the variance of the species multiplied by the selection gradient, the first derivative of these population rates with respect to the mean. So they're sort of you're moving in the direction of higher population level per capita growth. And finally, for the variance, we get the variance squared times the second derivatives of these two vital rates, basically characterizing, stabilizing, or disruptive selection. And then, because we have these this convolution in our beginning equations, we get an additional term, which is the birth rate times the mutation variance. So mutations are sort of constantly a source of variance for each species. And let us now take a look at one example of these equations. So Lotka-Volterra models, we have this trait density distribution, and we're going to assume now that the trait here is, for example, something like the beak size of a bird, and the birds are competing for differently sized seeds, where big-beaked birds are better at consuming large seeds, small-beaked birds are better at consuming small seeds. And then we have an environment or a resource spectrum, where we have sort of a lot of around trait 0, we have a high net per capita growth rate due to the environment. So we have a lot of intermediate sized seeds. And then sort of the further away you go from this, the lower your per capita growth rate is due to a lack of seeds, essentially. And WR is the half width of this function where you have positive growth rate. Then on top of this, we're going to have a competition between individuals. So everyone competes with everyone else. But the strength of the competition depends on how different you are from your competitor. So you're going to compete more strongly with someone with a similar trait than you're to someone with a different trait. Again, sort of allegorically, we can think of these birds competing more strongly with similar birds because they eat similar seeds. All right, so we have the machinery for tracking the density, means, and variance of any number of species. But we need to address sort of the main question of how many species we need. And to do that, we're going to try to assemble a community one species at a time. So let's start this Lotka-Volterra system with a single species. So we start here with a mean trait of minus 0.5. And because the optimal trait is 0, over time, the trait depicted by this line here evolves to become closer to 0. And the shaded area here is one standard deviation sort of standing in for the variance. And the density increases a little bit over time. And then the question is, once we've reached this equilibrium here, is this stable to the addition of further species? So in order to answer this, we're going to borrow a central idea from adaptive dynamics, and we're going to put in a very rare invader into this system. But because there is now a mean trait and a variance that can move around, we actually, once we put in the invader, we have to keep track of what happens to its mean and its variance. But we don't have to keep track of its density because we assume that it's exceedingly rare, so that the rates of the invader only depends on the density distribution of the resident and not of the invader itself. We write G hat for the net per capita growth, B minus M. And we can sort of visualize what happens to the invader over time according to these equations in a stream plot. I'll go over all the things that are here. So the arrow just indicates sort of the flow of these equations in the environment given by this resident over here. And we mark the resident with a plus sign, so it has a mean over 0 and a variance slightly below 1.5. And then we denote the stable equilibrium for the invader equations as a solid purple dot, and the unstable equilibrium for the invader as open purple dots. And this little rectangle here is just a zoom in of what's going on here around the middle. And then we can try to think, oh yeah, and finally, the green areas denotes where the invaders per capita growth rate is positive. So the question is then, what is the ultimate fate of an invader if you put it into this system? And well, as long as you sort of put it in outside of this little area in the middle, it's going to end up at one of these equilibria in the long run while it's still rare. And once it reaches here, because it has positive per capita growth rate, it's going to be able to invade the resident community, which here is just one species. So we can pick one of these points more or less at random. So here we have picked this one over here, close to -2, and insert it into the resident community. And once we do that, because its invasion fitness was positive, it will start invading the community. And once it reaches sufficient density, it will start co-evolving with the resident. But eventually, this two species community will reach a new eco-evolutionary equilibrium. And then we once again have to take these invader equations. And we can once again draw one of these diagrams. So now we have two residents marked by the plus signs here. And similar to before, we have three invader-attractor equilibria. All of them have positive invasion fitness or per capita growth rate. So we could put in another invader here. But this diagram actually shows something different compared to the last one. And that is that the resident equilibria now coincide with unstable invader equilibria. And essentially what this means is that this is actually a branching point, which means that rather than putting in a new invader, we can split one of the residents into two, and that and because it is unstable here, that should mean that they should diverge and branch into two new species. So let's pick this resident over here and split it into two. And if we do that, indeed, we can see that these two branch apart. And then once they have all of these three species have co-evolved into a new equilibrium, we can once again check our invader equations. And this time, all the attractor equilibria for the invader coincide with the residents, which means that the invader will have exactly 0 per capita growth rate or 0 invasion fitness, which means that once we have reached this stage, this community is uninvasible by any invader, and we have reached our evolutionarily stable community. Okay, so now we have all of this mathematical machinery. We can try to put it to use to answer these two questions that we posed in the beginning. So the first one is, what happens as the environment induces more disruptive selection? And the way we're going to do this is we're going to take our Lotka-Volterra model, and we're going to assemble stable communities for a range of resource spectrum widths. And it's well known from, for instance, adaptive dynamic studies that as you increase the width, you can cram more species in there. So we're going to take our framework here, that is the one with ITB, with intraspecific trait variation, and compare it to a framework without ITB, that is to say adaptive dynamics, and see what the differences are. So for small resource spectrum widths, there is not that much different. Initially, as you increase the width from being very small to being slightly larger, the intraspecific trait variation of the single resident increases. But at some point, the community becomes invasible, and we have a bifurcation into two species. But we can see that the dynamics are pretty, or the final community is pretty similar between when we include ITB and when we don't include ITB. And down here, we have sort of a zoom in on what's going on here. So the colored areas are these two species. The black bars here are the species that evolved when we did not include ITV. And then finally, the gray line here is a numerical solution of the full trait space equations, and the black dashed line is when we add up all the colored areas together. We can see that we have pretty good agreement between these two. So thus far, it seems that like including IDV didn't really do very much. We get very similar outcomes between these two frameworks. But as we increase the resource spectrum width, we start getting more and more different outcomes. So in both cases, we have an increase of species richness in the stable community as we increase the width. But the rate of increase is much higher when you don't include ITV. And as we can see here, without ITV, we get 6 species. And when we include it, we get 4. And as we sort of keep increasing the resource spectrum width, this effect becomes more and more pronounced. As we can see here, we get 5 versus 8 species when we include and don't include intraspecific trait variance in our dynamics. And it's also worth noting that when we do include intraspecific trait variation, we get quite different levels of trait variation within each species. So the central species has a lot of variation, the peripheral species have very little variation. So even if we try to use a framework with a fixed trait variation, as you sometimes do, it would not have been able to capture this outcome very well. So that is sort of question 1. So now we're going to try to move on to question 2, and what happens in structured populations, especially spatially structured populations. And we have we have derived these moment equations for the resident, as well as sort of derived how to carry out the invasion formalism. But the equations are a bit burdened, shall we say. So in the interest of time, we're not going to go over the generic equations for structured populations, and we're going to instead sort of jump to the 2-patch Lotka-Volterra model and try to answer our second question of what happens to trait variation as conditions become more different. So we're going to take the same Lotka-Volterra equations, but now they're going to take place on 2 patches. And the dynamics are identical on both patches, apart from the optimal trait being different between the 2 patches. So on patch 1, the optimal trait is going to be lower than it is on patch 2. And we're going to assume that there is some weak dispersal between these 2 patches, and that the dispersal is constant so that it does not depend on the trait. And then using our moment equations for the structured populations in combination with our invasion analysis formalism, we can, similarly to what we did before, we can take a range of different environments and assemble a stable community for each environment. So what we're going to do is that we're going to make the patches increasingly different from one another. So here all over on the left, we're assuming that the patches are exactly identical with a resource spectrum width spanning from -2 to 2 on both patches. And all the way over on the right here, the patches are so different that they have no overlap in where the species have a positive growth rate at all. So if you have positive growth rate on one patch, the other patch is going to be a pure sync patch for you. And let us first look at the outcomes when you don't include the ITV, that is to say, use adaptive dynamics. Because the outcomes are pretty simple in this case. Dispersal is quite weak here. So basically what we have 2 lines going diagonally down here, which is the outcomes that would have evolved on patch 1 in isolation. And we have 2 lines going diagonally up here, which is the outcome that would have evolved on patch 2 in isolation. Due to the dispersal, you get a little bit of bending in these lines, but that is basically what happens. And I note here that because we don't have any intraspecific trait variation, we're assuming that all the individuals within a species are identical, which also means that the black lines on patch 1 are exactly the same as the black lines on patch 2. However, when we do include intraspecific trait variation, different things happen. So when the patches are just a little bit different, we get a 2-species community rather than a 4-species community. But we can see that both the sort of orange and the turquoise species exhibit local adaptation in the sense that they have a lower mean trait on patch 1 and a higher mean trait on patch 2, and similarly for the turquoise species. And once again, we can look at zoom in and look at the actual trait density distributions here on patch 1 and patch 2. And we can also note that this local adaptation that these two species exhibit is in some sense symmetrical. They have approximately equal densities on both patches, and they sort of deviate from some kind of regional mean approximately equally. So local adaptation here sort of precludes more species from existing. But then as the patches become more different, the stable communities now will have 4 species, even when we include intraspecific trait variation. But as you can see, it doesn't exactly follow the case when we don't have intraspecific trait variation. So what we get here is basically we have a blue species and an orange species that primarily live on patch 1. And we have a red species and a green species that primarily live on patch 2. But they can also live on their off patch, primarily due to the fact that they can locally adapt a little bit on their off patch to sustain a significant density there. So here we have a sort of a different kind, an asymmetric kind of local adaptation going on. As we continue making the patches more different, we sort of go through a phase where we have 3 species, 2 peripheral species that exhibit basically no local adaptation, and a central species that exhibits symmetrical local adaptation, except around here at the exact symmetry point, where we'll have 3 species and no local adaptation whatsoever in any of the species. And then finally, as we continue making the species, the patches even more different, we initially have a little bit of asymmetrical local adaptation for the orange and the green species, as seen here. But as the patches continue being more and more different, they become sort of functionally disconnected almost. The weak dispersal just serves as a little bit of extra mortality on both patches, and what we end up with is a blue species and an orange species on patch 1, and a green species and a red species on patch 2, and they have virtually no density on the other patch. And they also exhibit virtually no local adaptation on either patch. All right, so what are we supposed to take away from all of this? Well, we have constructed this framework that lets us explore both the causes and the consequences of ITV under eco-evolutionary dynamics, especially for evolutionarily stable communities. So let's go back and try to answer some of these questions we posed in the beginning. So the first one was sort of this: as a practical matter, how necessary is it to include ITV to understand your study system or to answer questions about your study system? Sort of based on the theory and the models we have worked with, what can we tell people? Well, unfortunately, I only have this sort of non-answer to this. It's sometimes important. And the interesting thing is that even in our very simple models of Lotka-Volterra dynamics, we sometimes see that including ITV leads to different outcomes, but sometimes it matches up very well with the results when you don't include ITV. So it's you can't really give a simple answer to this question. Sometimes it matters and sometimes it doesn't. Similarly, how is trait variation apportioned in these stable communities? Well, we found cases where we had between species, within patch, and between patch variation, and all of these can be significant. But once again, it's highly system specific and hard to predict a priori. And then finally, how does ITV relate to species coexistence and richness? Well, I think it's important to be precise when you're asking these questions. So for example, when we increased the resource spectrum width in the unstructured model, we both saw an increase in ITV and an increase in richness. So if you sort of went out into nature and looked across this environmental gradient, you would see a positive correlation between ITV and richness. But I think perhaps more importantly is that when you compare the model and dynamics that include intraspecific trait variation, the final community will have less than or equal richness to the community where you did not include intraspecific trait variation. So this all sounds sort of a little dour, like I'm just coming with a bunch of non-answers here. But to a certain extent, I think that's one of the big takeaways is that you got to take the system specifics into account. And at least on the theory and modeling side, our framework can really help in deepening our understanding of the importance of ITV under eco-evolutionary dynamics. So thank you very much. The paper will be available very soon, hopefully. We just got proofs the other week. So hopefully in a week or two, you'll be able to read it all nicely typeset and stuff in the American Naturalist. And we were supported by an NSF grant, and we got a lot of good comments from 2 American naturalist editors and a reviewer. And finally, I'd like to shout out Sebastian Leon and 2 of his co-authors who started working on sort of very similar problems at the same time we did. And he was a very good sport about the whole thing. So go check out his paper as well in AMNAT, Multimorph Eco-Evolutionary Dynamics in Structured Populations. Thank you very much.
Thank you, Jonas, for the talk. If you have any questions, let us know in the participant list. Just raise your hand and I will call on you. So any questions? While people are thinking, maybe I can ask some questions. One of them is, this whole framework that you've shown builds on the idea that while you're rare as an invader, you evolve into your evolutionary equilibrium in terms of trait variance and trait mean. And that's fine. And coming from the same kind of modeling culture, we can stomach these kinds of assumptions very easily. But it is a bit strange, maybe, to say that in this case, evolution is super fast while population dynamics is relatively slow for a very small population. I'm wondering if there's just one thing one could do to auxiliate this and make it even better. Is it possible to add in the invasion phase some sort of stochastic extinction dynamic, some sort of branching process like calculation that approximates whether the population survives in the first place, whether it is able to get to the point where it dies? And if it can, then you just do the same thing that you've done. If it cannot, then you pretend that the species wasn't even born to begin with.
Yeah, so I've certainly been thinking about these things as well. And so one thing to start with, so like you sort of intimated a little bit, my initial thing was that this was like some weird backwards adaptive dynamics where the separation of timescale goes the wrong way. But that is actually not a necessary assumption for this. The only necessary assumption is sufficient rarity. You don't actually have to separate the timescale as long as the invader you put in is sufficiently rare in terms of density. The question is then, is that biologically reasonable to be like 10 to the minus 100 of the resident population? Like that is not going to happen even for microorganisms. So maybe, but mathematically, that's the way it works out. You don't actually have to separate the timescale. You just have to have sufficient rarity. And the reason it works like this is because, as you can see, these invader equations do not actually depend on the density of the invader in any significant way. So that part will not matter as long as they're sufficiently rare. So that's sort of point one. But again, like how biologically reasonable that is perhaps questionable. And the second question is sort of, what is the goal? So the way I've been thinking about this is sort of that the primary goal is to find this final stable community that is uninvasible. And this is a way of doing it. And then you can sort of think, all right, so this community, if you put in an invader here, it might die before it reaches positive growth rate and can invade the system. But if you take sort of the very long view and you think of like having a stream of invaders coming in occasionally, eventually one will invade close enough to positive growth rate to attain one of these attractors and start growing. Of course, the problem with all of this is that we have sort of completely thrown away any time information from this process. We essentially have to wait forever for this to play out. So amending this process with some kind of stochastics, like you suggested, might help us actually capture this whole process more correctly over time. Because that is something we cannot do now, which is why I put this weird scale the time down here. Like it's very unclear what the time scale of any of this is. Similar to in adaptive dynamics, where you have to wait for forever for anything to play out. So I hope that answered your question.
It definitely does. I was about to say that when you said that well, the given invader will arise, is that what you could use this to calculate is, or approximate, is the amount of time it takes for it to go. But so yeah, absolutely. Thanks Thanks a lot. And Chris has a question.
Yeah, and not so much a question, but more of a like follow-up on what you were just talking about. So I agree with Jonas that I think the way we think of this most, at least the way I think of this most, is just merely a way to calculate whether a community will be invasible or uninvasible. Not so much that you know, it's the actual dynamics of branching or what have you. That said, you know, if we're thinking about how like actual invaders would come in from outside the system, I suppose they probably come in, you know, with like as single individuals with clonal species, or a small group. So probably in the case of an actual bona fide like biological invasion, new species would come in somewhere on the bottom of this graph with low trait variance. So in that case, I suppose, like, you know, as long as they come in some region where there's positive initial invasion fitness, you know, in the green region, then maybe they'll be okay. But, you know, that's just kind of hand waving.
Can I actually make a quick comment on that? So how married are we here to the idea of mutants? Or we're forgetting the M word here. No mutants, it's all invaders.
Yeah, I very, very specifically used invaders instead of mutants because we already have this notion that each population has mutation going on inside it. So it would be, in a way, a little confusing maybe to call these invaders mutants as well. Obviously, they are potential invaders rather than invaders in a sense.
Thanks. Sorry, I'm not sure if you heard me. Liang, you had a question. Please go ahead.
Yeah, thank you. Thank you, Jonas, for the great talk and very nice, comprehensible figures. So I'm wondering, like, so in your model, have you considered the sexual reproduction? So normally, if we consider the variance in traits, the process of sexual reproduction would decrease the intraspecific variation. So they may this may not play a key role in a single patch model, but if you consider the dispersal between two patches, so that may decrease the adaptation in the other location if you already adapted to the local location and you want to disperse to the other one and constantly keep decreasing the fitness of your individuals in that patches. So have you considered about that?
No, we have not tried to work out how this would work under sexual reproduction. We have thought primarily about clonal reproduction here. To a certain extent, I think, and to maybe jury is better qualified to talk about this almost, because I know he has a paper where he did derive very similar equations using sexual reproduction. But I think the basic idea for the invasion formalism should be adaptable to the case of sexual reproduction as well, at least if you consider this sort of ODE framework where you always have an infinite number of individuals that can mate with each other, so that doesn't become a problem. But no, we have not looked at trying to extend the equations to sexual reproduction ourselves.
Okay, thank you.
To be fair, I think Geza is even better than I am, or well,
okay.
Much better than I am. What I can say is that when I was dealing with these quantitative genetic models, I always assume random mating. That makes it easy because the random mating always restores the normal shape of these curves. Therefore, you never get speciation, you never get multimodal curves like you do, Jonas. But Geza, I know, works on those cases where you get all of that stuff. So he can, I think, tell us about that. But before that, I believe Zach had a question. So please go ahead.
Sorry, I thought I unmuted, but I didn't. Hi. I'm thinking about your example of kind of the birds with different size beaks, and I wanted to ask, like, so when you have a multimodal distribution, you say you're treating them as like different species with separate normal curves, I guess. But but when I think of a normal Lotka-Volterra system without the variation, normally I'm thinking that the population of the growth of a species is like proportional to the density of that species. But when a trait changes, it doesn't necessarily make them separate species. So I wanted to ask, like, even when you have a multimodal distribution, are you then does the growth rate of one mode then not depend on the population of the density of the other mode? Are they really being treated as separate species? Because I can see how, like, they're strictly competing with each other now, but one mode could be helping the other if they're not really different species, but just like variations within one. You see what I'm saying?
Yeah, so the way we approach the species concept here is basically as a convenience to us, but the underlying dynamics doesn't really care about species. It only cares about traits. So you compete equally with anyone given that they have the same trait. Which species they belong to doesn't matter. So for instance, here we see that there's like a little bit of overlap here between these two species. So if you're an individual over here, you would compete equally with individuals over here, regardless of which species they belong to. What was that your question, or did you mean something else?
Well, I guess I mean, so you are keeping track of which species is which, even if their distributions are overlapping with each other, right? Yeah. Like an orange guy could be competing with another orange guy the same as a blue guy, but yeah. Say they could still reproduce with an orange guy, so the growth rate still depends on kind of the total orange. Is that correct?
Yeah. Well, I did write down the equations in case but they're a little involved. But basically, you have here. So this is the density of species I. And this is sort of the part that does not depend on anyone else. You have a basic growth rate, and then you have some discounting depending on how far away you are from the optimum. That depends both on the mean and the variance. And then this is sort of the competition. So you're competing with every other species, including your own. And it depends on the distance between your means and on the sort of summed up variance of your own variance, your competitor species variance, and the competition kernel variance. Did that answer your question?
Yes, it does. Thank you.
Okay.
Thanks so much. Giza, please go ahead.
So first of all, thank you for the very interesting lecture. I'm very interested in it. And then the underlying biological issue here, whether if we have a heterogeneity in the environment, spatial heterogeneity or niche heterogeneity, anything, it is covered by many species or covered by species with high variance. And in this clonal modeling, we always has a selection mutation balance. And I think it is generally true that if we go to zero with mutation rate, then we will find at the final state discrete species with zero variance. I'm not sure if you agree with me, but I think this is the generic picture. Of course, if you choose the fitness function very specifically, you can avoid this. But when you see sexual species, then it is more difficult because speciation of sexual species is not so easy. Evolutionary branching for clonals when there is no anything is needed, and mutation for the sexual population they need reproductive isolation, so it's not that easy. And what I'm trying to say that for clonal population, it looks if in the final state the mutation is only makes the situation worse. the exploitation of the resources were worse because the optimal situation is the completely discrete species. But in the sexual case, you cannot specify arbitrarily, so there is a more delicate balance between the speciation probability possibilities and the increased variance consuming the resources. So I find this very interesting issues, and we will talk it later if you want.
Yeah, so first let us take the clonal first. So I completely agree with your statement. So here is the graph here shows what happens when you change these mutation variants essentially. Yes. And these little arrows over here is the adaptive dynamics outcome. And not surprisingly, as we turn down the mutation variance towards zero, we get basically full agreement between our framework and adaptive dynamics in some kind of split of zero mutation variance. That should not be surprising if you have worked with adaptive dynamics before. But I think what is important to note is that it doesn't take a lot of mutation variance to sort of get a qualitatively different outcome. So I think like the mutation variance needed for there to be qualitative differences is not unrealistically large, even if in this example it is perhaps a little larger than would be strictly speaking realistic. Secondly, if you have a spatial structure, then you can have dispersal selection balance maintaining variance instead of mutation selection variance. Although that cannot happen in a single patch, obviously. And then finally, for the sexually reproducing species, that is something I must admit I have very little experience in modeling, and I can't really speak with confidence on what the differences would be. But I certainly agree that there are more things you would need to take into account to do it justice. So I'm not sure if I commented sufficiently or answered your question there, but feel free to clarify.
It's okay.
Thanks. Leon, you had another question. Please go ahead.
Yeah, thank you. Sorry for another question because I'm very interested in your research, and I used to work on the very similar topics. So I noticed that you also track the dynamics, the change of the trait variation. So I'm wondering if you have any like parameter values that generate like the explosion of the variance, and what do you do with that?
Yeah, so generally in a lot of these models, and sorry, let's see if I can find a good place to discuss this here. So often what you do, a lot of these moment methods revolve around Taylor expanding the rate functions. And the reason that it can be important is that you often get this sort of a quadratic term, which means that when you have disruptive selection, you just have divergence towards infinity in the variance term, similar to what you described. But in principle, if you integrate across this normal distribution, if you have any sort of normal rate functions that you would normally use in modeling of the ecology, then this prevents the blow-up from happening. So we don't really get variance blow-ups in this formalism unless we have to Taylor expand the rate functions. So it's very hard to get the variance blow-up from happening here without specifying some rather strange rate functions. Or did I misunderstand your question?
No, it makes sense. Yeah, thank you.
Thanks. Are there any other questions? While people are thinking, this is very good for demonstration, and actually it's perfectly good. So I'm just going to ask this question I'm not sure. And anyway, I'm not sure what your thinking is, but are you thinking of extending this framework to other kinds of competition than Gaussian, or that's not of interest because Gaussian competition already contains all the essential components of?
Yeah. What we have not done is we have not looked at other distributions for the species other than Gaussian, if that's what you mean.
I meant more the competition kernel, or the
yeah.
So, for example, we have competition colonization style hierarchical competition.
Yeah. Actually, when we started this project, we did not have Lotka-Volterra competition at all. We did explicit resource competition instead, and you can actually find in one of the appendices of this paper. We have a stage structured model where there's competition for two explicitly modeled resources. So this sort of the framework itself does not rely on Lotka-Volterra. It can be adapted to most common things, similarly to the way adaptive dynamics, you sort of plug in your things and you can see what the eco-evolutionary sort of dynamics of that thing is. And like I said, we looked at a few cases of explicit resource competition, and it works pretty well. The reason we gave it up was partially because it was so difficult to explain what was happening, and partially because it was hard to find in a couple of those examples. Getting a good illustration of the difference between intraspecific and interspecific was kind of hard to achieve in any meaningful way. So yeah, you can certainly use the framework for more complicated models than Lotka-Volterra, and it's mostly plug and play with a possible with one sort of caveat, and that is if you have these simple functions. So, for instance, polynomials, exponentials, Gaussians, you can evaluate these integrals analytically. But if you have, say, a type 2 functional response, you cannot. Which means that you either have to do numerical integration, which is possible, and you can do that quite efficiently using Hermit polynomials, I want to say, when you have a integrated
when you have normals, that is Hermit. Yes, that's the natural
so you can at least in one dimension, doing these integrals numerically actually requires relatively little computational power. When you have a higher dimensional trait space, it's worse.
So this is the reason part of the reason why I was asking. So recently we made the discovery, and take it or leave it, I don't know if you're interested, but I always thought that if you have a sigmoid function describing some sort of hierarchical competition, you cannot efficiently do these integrals. You might be able to do one, but when you have to do the double integral, it fails. No, you can do it. Mathematics,
yeah, you can do it
that you can do it. The Bronstein giant table of integrals doesn't know that you can do it, but there's some very obscure publication in an integral table from the 60s. I can send it to you.
Send me that one. Yeah. So you can do sigmoid. So what I find out, and I think this is known to a certain extent, if you use the primitive function of a normal distribution, like the error function, that is a sigmoid, and that one you can integrate analytically. Although
that's what I meant. Yes. Okay. Right. So you know that already. Okay.
Yeah. But a normal type 2 style sigmoid or like saturating function, you can't, as far as I know. But
I think you can, at least some types, weirdly.
Yeah, so that is certainly a potential problem when using this. If you have simple functions and you can evaluate these integrals analytically, it all works very smoothly. Yep. If you can't, you either have to do numerical integration or you can do Taylor expansion around the means to whatever order is necessary. And with like modern computer software, the computer can do the derivatives for you. So you don't have to like write down the 5th order derivative by hand. So it's not that bad, but yeah, it's the Lotka-Volterra is idealized in the sense that you can evaluate all the integrals over the rates analytically, and typically you would not be able to do that.
Yeah. But from my personal experience, the computer really struggles if you have to do this integration numerically. and yeah, okay. Well, I think we're getting a bit too technical here, but there's one big problem with the Hermits polynomials, which is that all these quadratures only work really well if the Gaussian function is integrated with a polynomial. If it's integrated with another transcendental function, you're out of luck. You might have to use so many sampling points that you're better off trapezoid.
Yeah.
In integrating. We were very happy to discover that you can do this sigmoid integral, because the computers were really struggling. It also depends on how many species you have, of course, but if you have a couple of dozen, then it becomes too much. But anyway, I digress. Any further questions? If not, then thank you again for the talk, Jonas, and thank you everybody for coming. And I'm not sure when our next seminar is. Do we actually know when our next seminar is supposed to be, Chris or Geza? Do you know?
Yes, I know. Just we need to be sure. The next lecture is March 7, Jürg Spack. And the following one is March 14, Christy Hernandez. So we have a longer period without lectures, but then after that 2 lectures from 1 week distance. So 7 and 14 March.
Thank you, Geza. So 7th and 14th of March. So see you there. Sorry, Chris, did you want to say something?
Have we missed Caroline Farrier then? Because she was going to be on the 28th.
She quit. Yeah. She
That's happened. Okay.
Cancelled.
Yeah.
Hopefully she will give it at some later time.
Yeah.
Okay. Anyway, thank you all again, and see you March 7th then.
See you.
Bye, everyone. Bye-bye.
Hello.